Okay, it is possible that we are live, boys and girls. We don't know. It's a hypothesis. We think we're live. We don't know. Anyway, this is the after show part 2 to last night, and I just want to say a few things about last night. Number one, certain people, fuck you. I'm a little bit not particularly happy with things turning into dumpster fires and people. And it happened yesterday on the weather report where people were being a little pedantic about definitions, and I'm going to start throwing people in the back room. And if you get mad at me and you decide not to come to my show anymore, I guess I'm going to have to put up with that risk, because I notice like we'll have 25 viewers, and then it will turn into a pedantic bullshit dumpster fire over some little mutant point that has nothing to do with what we're talking about, nothing to do with science. And I'll watch our viewers bleed back down to 11, right? And that means that nobody's enjoying this shit. Not me, not those of us who come here to actually talk about stuff, and not the audience, So what the hell am I doing this for? And last night, it was great that we had the creationists over. And we could have had a number of discussions about biology and religion. I don't know, but none of that stuff happened. So today, along with that, I've had about 3 or 4 hours of sleep. I got to sleep at 7 a.m. because you fucking young people kept me up all night.
Damn young people on their parties late at night.
Yeah.
Jeez. Yeah, I saw the after show went on for like 3 hours. I was like, dang, Steve, what are you doing?
Well, not only that, but then afterwards, I was warmed up and I went and watched TV, and those guys, I think John and Argatha and what was the other guy? Redefine Living went at it for another, and Snake went at it for until 7 a.m. And I kind of wanted to end the stream because my computer burns into the ground if StreamYard is running with an open stream. And I don't want to ruin a $5,000 Mac, So I stayed up and watched it and talked a little bit at the end of it, and then we went to sleep. But I mean, it was cool. We got like 150 views, which is more than I normally get. I normally get around 100. So I mean, thank everybody for that. But 150 views isn't exactly something to write YouTube about,
Right?
Request the medal. And we'll never get any higher if we don't do something a little bit different. So I don't know.
Yeah. I would just say StreamYard, I think of it as a privilege, and if you're not going to behave, then you probably shouldn't be in it.
Right. Yeah. I'm just going to have to be better with the backroom stuff. And I found out how I can ban people from the studio. I didn't think I'd have to ban anybody, but damn, Jason Ezard showed up. And then he wrote me an email threatening me with, I know things. It wasn't even a full sentence, but basically he knew those things about me. It's like, well, I think he was trying to say I know things that nobody else knows. That's like, no, you don't. Because I mean, I'm the kind of person, I'll go to a grocery store and somebody say, how you doing? And I'll say, yeah, today I was having this like psychological issue where, I was like, I wanted to leave my wife, and about a half an hour later, I mean, I tell everybody everything about my life, right? I'm very, very transparent about stuff. So right. I really doubt he knows something that nobody else knows, because I just blabber. Okay. So Standing for Truth came in, and he wanted to go on, and we did start to get into genetic entropy, but we didn't do it in a manner that would work. And you guys didn't get into it in your talk, right?
Yeah, not really.
Yeah. But what you did get into, phylogeny. So I don't know. Andrew, what do you think? What do you think of your talk last night, now that you've had like 12 hours to sleep on it?
Right. Let's see. So I was looking over it this morning, and I think he brought up a good point that I, had thought about, but I hadn't really taken into consideration, and I wanted to hear your guys' take on this. The fact that phylogenetics, as far as I can tell, it is correct that cladistics, I'm just going to call it cladistics, they're synonymous. The fact that cladistics assumes, universal common ancestry, that it assumes that there's some kind of relationship. And
Right.
what I was trying to grapple with is, isn't it also true that, for instance, isn't it also true that any hypothesis has to make assumptions? Like
Yes.
even theories. I mean, like you got to make assumptions. You got to assume, for instance, your theory is true, and then see, okay, if X is true, if and only if X is true, we should see Y.
Right.
We see Y, therefore X is most likely true. So like you have to make assumptions. So I don't know, what do you guys think of that kind of point?
It was odd that I forget how he did it, but he was kind of pushing, there has to be universal common ancestry, which is kind of like building a straw man in a way when it comes to phylogenetics, because you pick a proposed, outgroup and a root of the tree, right?
Right.
You don't have to pick a root of the tree. You can just take a look at where the phylogenetics takes you, and you have an unrooted tree. So
yeah, you look for relationships, but then it depends on whether you want to take that step further and use an outgroup and look for the root of the tree, basically kind of take the relationships and also put oldest common ancestor to it. I'm trying to think of my words here.
Yeah, you're looking for a relationship, and you're kind of looking for the length of those trees, those branches.
Right.
Yeah.
Like in parsimony, for instance, which is kind of what I went over. You're looking for the tree with the shortest length. Basically, you're looking for the tree with the fewest number of steps in total. Steps in this case just being, okay, state A turns into state B, or A prime or whatnot. So you're just counting up those changes that you hypothesize and saying, okay, which one is most likely correct based on this optimal criterion of parsimony, basically. And which I kind of found interesting when I learned about this. I had always assumed parsimony and things like that were used to actually build the trees. No, it's actually used to assess the trees, to figure out which one. Yeah, to figure out conflicting. Because like if you take one trait and you ignore all the others, and this was one point I was trying to make, is that you take one trait, you ignore all the others, you're going to get a tree, but it may not be the correct tree because it doesn't agree with most of the evidence.
Yeah.
So you have to take all the character changes into account. And this is what I was trying to point out with cars, for instance. You can create a nested hierarchy. You can use cladistic methods on this, actually, which I also found interesting. But it doesn't necessarily flow from the data. So you can use those kinds of methods, but you're not going to get as neat a tree as you get in life. And like, sure, there are incongruencies, but that's just because we're not perfect samplers.
Wouldn't it be interesting to get a whole bunch of cars, like modern cars, like 2020 vehicles, and do some phylogeny on it?
Yeah.
List all the features of the car?
Yeah. The cladistics of the car.
Has anybody done that? Because it would be interesting to see if you actually can do that and how it would come out.
Yeah, that would be pretty interesting. Let's see. All right. Nestlig here says, polytomies sometimes happen, but they're relatively rare for the most part. Yeah, I think that's true.
Yeah. And a polytomy would look like what?
Okay, so say you have 3 species. Now, normally, sometimes you can find the relationships between them, which one is the sister group to the others, basically, which one's the outgroup, basically.
Right. You'd end up with 3. It's one branch with 3 or 4. Instead of a binary tree, you'd have, multiples.
Right. You'd have a node and then 3 branches coming from that node, basically, in that example. So it's a case where you can't determine which is more closely related. You're just going to say, okay, let's wait for further evidence.
Right. Yeah, and I think with cars, I think you'd come up with a mess, but I don't know. Maybe there is some really good way, and we could put in aquatic, all these military vehicles and all the cars that they love to show, and we can figure out what in the hell is up with the Ford Pinto.
And would Hot Wheels count as cars?
And Hot Wheels, yeah, I guess
Hot Wheels. Yes.
Well, they'd be almost like prokaryotes, wouldn't they? Sort of.
Oh, that's a good one.
I think we should do this.
Yeah, that would be fun.
And go-karts.
Oh, go-karts.
Yeah. And, and horse and buggy, of course, we've got to go way back into the pre-Cambrian.
Trains, subway trains.
Trains, there you go. I mean, I wonder how it would all turn out. I don't know.
Yeah.
And let's see. Okay, I like to show someone. I like how someone actually attempted to do classified dragon.
Raw.
Really? And it became a mess?
I think he tried dragon taxonomy, but I didn't see how it worked.
Nestle can't bless us with his presence today.
Oh. Well, thanks for at least commenting, Nestle.
Learning synaptomorphies or synapomorphies and apomorphies and all the other morphies, that is also very challenging.
Yeah. Like I was saying last night, what is apomorphic and what is plesiomorphic depends on the group you're studying.
Mm-hmm.
And I gave the example of that with vertebrae, and I don't know if he really took that into account. What a point I wanted to make to him, which I didn't get to, was basically what you mean by derived. Sure, you're assuming, quote unquote, an ancestral relationship, but you can clearly see that kind of relationship in the data. Basically, you can see that, okay, these things share this unique trait. Nothing else has this. Then these three share this unique trait. Nothing else has this, etc. Now, obviously, that's simplistic because there are many, many characters you can include. But I like to use the word uniquely shared traits rather than derived traits because it shows that the relationship is assumed, but it's also not assumed. You know what I mean?
Yeah.
Like you're assuming there is a relationship, but you're not assuming what the relationship is, basically. You let the data speak for itself. Right.
And then Stanley brought up that paper. He brought up that little thing from ICR, which I had ready last night, and I wanted to talk about it. And they linked that paper by Ettersberger.
Right.
And I went and looked at that paper, and he was talking about a problem with phylogenies when we do it genetically, that we come up with more than one tree. You come up with different trees, and sometimes it's very hard. And I think he was claiming that, and the paper too says that on some genes, it looks like we're more closely related to gorillas than we are to chimps. And part of that is because of incomplete lineage sorting, which and I'm a student myself, but that is when you basically two populations break, and one population can have a certain subset of genes because of genetic drift, and the other one doesn't have them. So you'll end up with kind of an odd break, where if you look at those particular genes, you may like humans and chimps may have broke off, and when the humans broke off, they took part of the gorilla genes, and the chimps did not.
Right.
Does that make sense?
Yeah. That kind of like that's why you kind of need to involve lots and lots of characters, basically, because it's like the more characters you include in your analysis, hopefully the better the tree becomes. It usually does get better and better over time. No one disputes that that birds are part of the class Reptilia, quote unquote. Well, maybe except creationists. I don't know. This was the other thing I found confusing, was he was saying, okay, you have this tree-like pattern for all life, but you also have a tree-like pattern that stops for all kinds. And I'm like, what? How does that make sense? Can you know what he was saying by that?
I can't remember that part. Yeah, okay, repeat that again. Let me see if I can.
Okay. So he showed a picture of the kinds, right? Like these tree-like patterns, and then it stops basically. So individual trees, separate creations, basically.
Okay.
But then he also said that something along the lines of that you would see a pattern further down between the kinds. And that doesn't really make sense that you would see this kind of relationship basically for the kinds, but you say there's not a relationship, a genealogy basically for between kinds. That doesn't really make sense. That's kind of arbitrary.
Yeah, I'm not sure entirely what he meant.
Yeah.
I'd have to listen to him again. The thing is, you're coming up with, you're bringing evolution or common descent to the table, or you're bringing trees to the table, and Standing for Truth's job is to shoot enough holes in it that it looks like the Tin Man is going to fall down. That your man is going to fall down, and that's kind of the way he operates. So you have to be careful with him, and he did start to really gallop on you toward the end.
Yeah.
He started bringing up ERVs and paper after paper after paper, And
Yeah, I was getting kind of, I was kind of too tired to really stop him there.
Yeah. Yeah, and then last night we got into bacteria and mutation rates, and I did some numbers on that, and of course we got into genetic entropy, but I guess that's outside this discussion. But what the hell? I ran out of paper.
Uh-oh.
That's weird. I ran out of paper. I didn't think you could run out of paper on this thing.
You ran into a singularity.
Something's wrong, because I ran out of paper. I put a picture at the end here, and I ran out of paper. I don't know. Interesting. Well, damn. Can't have that. But yeah, I'm not sure what he meant by that. But the devastating. Well, okay. So if you run phylogenies on multiple genes, you'll end up with multiple. You can end up with trees that are slightly different one from the other. If you're just looking at one gene, or if you're looking at like the D loop or some non-conserved portion,
right?
Like the one they use for barcoding, that CO1 gene, and you'll end up with differences, and they consider, oh my God, that's a problem for evolution. No, not really. Because of genetic drift and because of which genome you happen to have. I mean, there are going to be all sorts of differences. The thing is that when you run enough of them, and I want to start doing this with Mega. I don't know if you're interested in learning how to use that Mega X.
Right.
And do some, Cell, let's see, Cell and Erica. Erica used RStudios, which you can do some work with.
Okay.
And Cell was using Mega, which is the creationist favorite. But I would like to run some. I'd like to run some different genes. Particularly, I would like to run DDX11L2, that chromosome fusion gene, and see. There's a couple things you could do. I mean, you can even run, like, I think you can do like a phylogeny of a protein, where you can take one genome. You're not looking at multiple genomes, but you're looking at similarities between one protein and other proteins in the genome.
Right.
So there's ways to do family trees for proteins, because something that nobody ever mentions, creationists, and we fail to mention it too, is that proteins come in families. And you'll find one protein that has a whole lot of homologs that are very, very similar to it in one genome, in our genome.
Right. And that's due to duplication, right?
Yeah, yeah. And we can see that all over the place. These protein families, like G protein coupled receptors, there's 700 of them in our genome, and they are very similar. And they all have what is it? 7 MT? I can't remember. They have 7 alpha helices that sit inside of the cell membrane. And they have an outside, and then they have an inside part. And the outside part recognizes a signal, and the inside part connects up to a G protein and turns it on or off, turns it on. That sort of thing. So there's 700 of those guys. And it would be interesting to look at those families and do phylogenies for those. And I'm not sure how much work has been done on that.
Yeah. I think Nessa just said something interesting in the chat, which I think was related to what I said. Let's see, baraminology. That's the thing.
Okay, so let's go.
So baraminology, and he said that if that's what I was saying, Nessa, basically, that like if you were to find if creation were true, you would expect to find polytomies near the base of this universal tree, but you wouldn't expect the hierarchy to go down that far. And that's kind of what I was confused about last night, was that he was saying okay, the tree-like patterns within the kinds, but the tree-like pattern is also between kinds. Well, it's just really confusing, because then if there's tree-like pattern in kinds and that shows relatedness, why doesn't the same pattern show relatedness between kinds?
Yeah,
like it's special pleating. Is that the way it's called?
Yeah. Well, I call it a rescue device. They have this.
Right.
They have these rescue devices that they claim, okay, when we do run into something that looks like a tree, they'll say, well, God did that for a purpose. We just don't know what it is yet. It could be that the organism needs that difference in its genes because it may crawl off into a different environment and it has to turn in. So I don't know. So that's one of the rescue devices for finding these deeper, more intricately connected trees, which should not be the case. The created kind could be completely different.
Right. That kind of ad hoc reasoning is okay if you want to build a hypothesis, but you still got to go out and test it. And they haven't tested a darn gone thing.
Yeah. And the other thing they can't account for is in the non-conserved regions where the sequence does not matter, where the only thing that really seems to matter is the length of the sequence, like in introns and things like that, which amount to 27% of the genome. There's no reason why there should be a phylogeny in those areas. And yet that is exactly what we find. It looks exactly as if we came from a common ancestor and then different branches mutated to varying degrees,
right,
over varying amounts of time. There's no reason, and I always describe that as God didn't need a sequence, so he just penciled something in really fast, and then the next time he came along, he penciled in it again, but he made mistakes every time he did it because he was distracted by. I think he was distracted by Eve, to tell you the truth. I would have been, but anyway, so he kept making more and more mistakes as we went along, right? And as we go, closer to the present, he made almost as many mistakes as you would expect to have if we all descended from common ancestors back on the tree at different times, right?
Right.
Like instead of at one time. I mean, it should have been those mistakes in the non-conserved regions, I think, should have been identical in every kind, in every species. There's no reason why they should vary in the amount that they differ, because you're making a mistake,
Right.
I don't know. Does that make sense?
That's a good point, that like these these methods they're using are kind of unfalsifiable in a sense, right? Like they're consistent with the data, but sheer consistency isn't by itself good in science.
Yeah.
It's kind of complicated to differentiate between the models, but like saying God did it isn't a scientific model to begin with. I don't know. What are your thoughts on that? How do we differentiate between these models, basically?
Between their model and ours?
Yeah, like how do we find which one is, because they would say it's it's just as consistent as common ancestry. Now, obviously, I would disagree with even that, but granting that, how would we differentiate between them? I guess is
Yeah.
the question we need to pose to them.
Yeah.
And of course, from there, they would go the junk DNA, right?
Oh shit! What did I do? I got rid of one of my exons. Got rid of an exon. I want to put an exon out here. So I got an exon, and I got some introns. These introns, are not highly conserved at all. There's like a pivot point that allows it to splice, and there's a few other signals. I think there's another signal here that's upstream, and that allows it to splice out the intron, and this is the exon. So we have exon, and this is an intron, right? Do you follow that, Scott? Are you with us, Scott? Scott's muted. So anyway, so we got a highly non-conserved region here, and if you look at it, it's
Only time you ever call my name is when I go into the kitchen to get something. So you have diagram, and this is
you can't see my G.
non-conserved regions, which means what? Genetic code that is not passed on, or
no? There's if you looked at the mutation rates in the non-conserved regions, you'd find that they mutate a lot faster. When you go from one species to another, there'll be more differences in these introns than there will be in these exons.
Gotcha.
Because these exons are what makes our perfect little protein machine. With beta sheets and stuff like that. And it has to be, and say we have like an active center right here in a bunch of alpha helices and a couple more beta sheets. So we have an active center right here that has to be conserved, right? I don't know who bioluminescent brain is. Could somebody tell me who bioluminescent brain is?
Oh, is he in the
trying to get in?
He's in the studio, yeah. So, Dr. Dan is here to straighten me out. Anyway, so you have that difference, and there's what the heck did I just do?
Oh, it's brain bug.
Oh, okay.
What bioluminescent something?
Hey, brain bug.
What's
is it
up?
brain bug?
It is.
Hello, bug.
Hey, brain bug.
So,
How's it going, guys?
it's going good.
So before we get into this brain bug, what did you think of my discussion last night with SFT?
I did not see your discussion with SFT. What I miss?
We were talking about whether nested hierarchies fit more with design or common ancestry.
Oh, good. That's the debate. So you talking? Was it a genetic discussion, or are you talking dispersal, or what all did you cover?
More like the methods of phylogenetics, basically, and all the assumptions involved, quote unquote.
You'll have to go back and dig into that. I had family stuff last night. Sorry.
Oh, yeah, that's okay. Oh, there we go. Yeah, I think creation myths. Dr. Dan made a good point. He said that nothing is falsifiable in terms of God did it, in terms of design, but plenty of things would be falsifiable for common ancestry, and that's the difference. Basically, why are we going to go with a model that is consistent with anything? Basically, I think that's a really good point.
I'm trying to go through his chats here. But anyway, these non-conserved regions will have a lot of changes, and these change, like in a species that we think is close to us, we might have an exact copy of the thing, except for like one nucleotide, right? And then in a species that we think is more unrelated to us, we'll have we'll have some homology, but we'll have like I don't know, we'll have that, right? So this one is obviously closer to us. Well, why did God do that if this nucleotide sequence has no meaning at all? And they will say, oh well, it needs this particular intron has got to basically it needs to have that in order to be a cat, and it needs to have that in order to be a dog, and that in order to be a wolf. So they'll come back and they'll also say that basically if we don't know what it's for, it's just because we haven't found out yet, because they will claim that the genome is 100% functional and there can be no junk in God's design, that God meant to do this. And I suggest that maybe God made mistakes, that maybe God was sloppy about these areas that he didn't, that weren't conserved. And, maybe he had a bottle of wine and he made, so many mistakes here. And as he drank the wine, he made more mistakes in this lineage. I don't know. It's a hypothesis. But Dr. Dan is right. There's really no way to test that, because they can make up any shit they want that points to God did it. And they can come up with, that's why they came up with created heterozygosity. And when that didn't work, they came up with, what was it? The they claim that Eve that every egg in Eve ended up with a different genome, right?
What?
So it wasn't just, so now we have more alleles available. We got like 30,000 of them, and they'll come up with one story after another. So you can't really plug that dike, So there's just no way to battle them other than to expose their lies and expose, they will come at us about abiogenesis and they'll say, oh, well, you don't know how it happened. You don't know. 70 years, you don't know. And then 5 minutes later, they will say, well, we don't know that those non-conserved regions aren't important. But That's okay. It's okay for us not to know when it comes to non-conserved regions, but it's not okay for us not to know about the stuff that matters to us.
I coined the term a couple of weeks ago, and it's still a good term. It's called genomythology.
Oh, yes.
They're just those stories made up to explain what they don't understand or know about genetics.
Right. And they can do that all the time. And they can come up with just those stories. But if we come up with any kind of a story, even if it's backed by evidence from the lab for abiogenesis or the eukaryotic cell pop, that's not okay. You see the double standard there?
Oh, yes, definitely. And what does Otangelo and others who say this expect the scientists to have done within the last 70 years in terms of abiogenesis?
Oh, they think that's long enough to go back and do forensics that are 4 billion years old, and that we should have found something already. And then and they admit that even if we do find something, they're going to claim, oh no, an intelligence did it in a lab.
Yeah, and also to say there's been no progress in that area is just insane. Whenever I followed up a quote that Otangelo copied and pasted, I found that the guy that he was quoting actually was writing his paper to show that there has been progress in abiogenesis. So, it's just he only quote minds, reads what he wants to read into it, and then doesn't read the whole paper or its context.
Okay, why there isn't a DNA bottleneck in all animals after Noah's Ark? He mentioned something about, do you need barcoding in some article by Rockefeller? Okay, I'm not aware of the Rockefeller article, but yeah, they came up with, Erica handled the barcoding thing pretty well the other day in her epic 3-hour broadcast, I think it was, where she went through all that.
She has spent considerable hours lately busting creation science.
Yeah. And the thing about the bottleneck, like the out of Africa bottleneck, they imply that that we came up with this recently as a just so story to make things up. And I don't know, I swear I was hearing about this bottleneck like 30 years ago, 40 years ago. I mean, we kind of knew that if we came out of Africa, there had to be a bottleneck. I mean, there had to be a bottleneck in this transition to Europe. And, we may have had multiple little spurts of groups that came out of Africa, but they weren't too much bigger than a few hundred people,
Right.
So, I don't know. And then he
Yeah.
claims, oh no, that's a real problem. If the bottleneck was 10,000, then interbreeding would kill us, and that's simply not the case. A
Right.
population of 10,000 is pretty damn good as far as genetic diversity, as far as I know. So, does anyone know what he's talking about? DNA bark. Oh, Jerry, are you not familiar with the whole DNA barcoding thing? So they came up with a region, the CO1 region, which is highly variable. It has part of it that's conserved, but there's part of the gene that is non-conserved. And it turns out that in every species, there are mutations in this area. So it becomes a really good way for you to barcode something and tell what a species is. So what they dream of is you take all the animals in the forest and you put them on a conveyor belt, and a clerk, grocery store clerk, barcodes them and tells you, this is a this, that's that, this is this. That was the idea behind barcoding. To the best, and I could be wrong, but I believe that's what they were up to. Is they were trying to find a really good way to find out, to differentiate between species. And they came up with this one little portion of a gene, and then Raw Matt takes it and he goes places that no one should go.
So each kind should have its own unique barcode that we should be able to look at its genetics and see the barcode in its DNA and say, this is a fox and this is a weasel. And
right. Yeah. And what they found is that it seems like 200,000 years ago that every species kind of developed its own unique barcode.
That doesn't help a 6,000-year-old Earth, though, does it?
No, it doesn't help. Well, they kind of slide over that. They use the same evidence that shows 200,000 years, and they insisted 6,000 because of course they use what is it called? My brain just lost it. Pedigree, which you can observe, right? And we use assumed mutation rates, so we make an assumption as to how long it was. Anyway.
Hey, Speed, since Dr. Dan's in the chat, you mind if I shout out a question for him?
No,
related to the discussion?
He was having a lot of.
All right. So, Dr. Dan, if you're still there, would you mind explaining your thoughts on if evolutionary phylogenetics assumes genealogical relatedness a priori, or if such an assumption is actually evidence for common ancestry, if that makes sense? I just want to hear your thoughts on that if you're still there.
While you're waiting for his answer, what channel was your debate on? Was it on Steen's channel?
Of course it was. That's the only place he'll do them.
He won't go on. Well, I think he goes on Modern.
Well, actually, he came on my channel last night for the first time, I think, ever into one of our discussions, and I guess I shouldn't slam him too much for that, because they did show up last night, and I got 50 more views than I normally get. Okay, we're going to Nestle. What the hell are you doing to us? Okay, we asked Dr. Dan a question. Nestle is coming. What? Nestle is writing a book.
Yeah, or at least a paper.
Yeah,
this is probably Nestle's area of expertise, I'm guessing.
3 species with an A and one with a T. You cannot tell which. Oh, yeah, I think he's talking about how some of the data gets lost in some of these conserved regions. I'm not sure where he's going with it. I'm really tired today. Is this Dan's answer to your question?
I think so. Let's see.
Phylogenetics is evidence for common ancestry. We make predictions that must be true if they're related, and then see that those predictions are accurate.
Okay.
It's pretty square to me.
Yeah.
Oh, so Scott, introns and exons, you wanted to know what those were?
Well, I've looked it up, and I found a video from some biology teacher from California, so I just thought I'd paste the link to it there in live chat.
A biology teacher? What if it was Mr. Batman?
Yeah.
Oh,
right.
no. Yeah. I didn't see anything about pedophiles or whatever.
Yeah.
Oh,
Lena just corrected my spelling, and I appreciate that, Lena.
Can anyone think of a random gene? Random gene. Random
Gene Simmons.
Gene Siskel.
Why? Oh, you guys. Random
awesome.
gene.
That was
Random.
awesome, Scott. Glad we're
And
both
don't
on the same
forget,
page.
don't forget the best of all genes, Gene Roddenberry.
DDX11. Maybe how about DDX? DDX2. Let's look at a DDX2. Zero domains, genes. Okay, genes. Come on. There are 7 of them. That's all? Are you kidding me? I'm looking something up on NCBI. Okay. Initiation. What in the hell is happening? What's happening? Anyway, you guys go on and discuss.
When you really try to think of any random gene, is it just me or is it everybody who the first thing that pops in your head is the Sonic the Hedgehog gene just because of the name of it?
Oh yeah, Sonic Hedgehog. Yeah,
Yeah, that's the very first one that comes to mind.
everybody loves that devil. The person that named that should get the Nobel Prize for naming genes.
Yeah. Have you seen it? Looks like, I mean, it's apt. It's a good name.
Yeah. Okay, so I'm trying to find a gene in a database, and I have to. Why am I having so much trouble today? Okay, genomic contact summary. Where is? Oh, there it is. Okay.
I did get to see Dan, Dr. Dan meeting Danny yesterday, though. That was entertaining.
Oh yeah. Oh my God, yeah, those two just tore into each other, and it's
Yeah. Are they going to come back and talk like on your channel?
I think so. Yeah, I'm hoping so.
Yeah.
We're also trying to hook this philosophy of math thing up, but Leo and Danny are like herding cats. And God, it's probably going to be nothing but a fight over, definitions.
That's what it seems to, from what I was listening to, it really it comes down to the terminology, and everybody's not on the same page with the words you're using. So how can you have a conversation about the subject matter at all if you don't use the same language?
Yeah.
But,
okay, so here's a gene. I believe this is DDX11 or DDX2. And the name of the gene is something else, which I forgot. But okay, you see those little green bars? Those are exons. And the skinny lines in between are introns. So our genome is about 20% introns. And what happens is inside the nucleus, after that gets transcribed from the DNA into an RNA, it's called, I think it's a pre-mRNA. Then these sections, these little lines are spliced out. And sometimes it'll, and you probably can you see my cursor? Okay. Sometimes it will splice out just the intron, but sometimes it will splice out the intron and a couple of exons, and you'll end up with 2 different proteins from one gene, that sort of thing. But the only part that ends up turning into a protein are these exons. So the others, and this only happens in eukaryotes. And that's one of the reasons why bacteria have a much smaller mutation rate, because they have more conserved DNA. They have a higher amount of more conserved DNA. They don't have introns. That is a eukaryotic feature. And there's a whole lot of work done on why in the hell do we have introns.
So introns are 1s and exons are 0s?
What?
Like binary, where there's you got nothing, and then you have
the exons would be the 1s.
Okay, the exons are the 1s, and then
yeah exons mean they're exported, that they get outside. I think that's where it came from. And introns is they're kind of the intermediate stuff in the middle that doesn't matter. So we have Demeter trying to enter. He's gonna straighten my ass out, hopefully. But anyway, so let me see here. So we can go minus and take a look at other genes out here. We have that gene right there that we were looking at, and then we have other genes out here. And you can kind of scroll along And you can find all kinds of these. And look at the introns. These are non-conserved or not as highly conserved. And this little guy is packed chock full of exons. And you can just look at all these different things in, I believe we're in chromosome 7, is it? 17. So we're in chromosome 17, looking at the whole picture. And you can go further out and look at even more stuff. And these are variations, I believe, going up and down. They have across the population, there are differences in some of these sequences. So they will list all the different, all the variations, all the alleles that pop up. And I'm not sure what I did there, that sort of thing. But anyway, so you can keep going, or you can go out to here on one of these. Oh my God, I shouldn't have done that. Let me go back. Because, okay, so let's try to get into an exon here and take a look at what is that. Okay, it actually shows you the actual sequence in the DNA. I mean, it goes out that far. And unfortunately, when you go out that far, it's very hard to find your exon again. Oh, there it is. Okay, so now we're looking at the exon. Okay, so now we're looking at the actual sequence. And then on some of these, I believe it'll actually show you what the amino acid is that gets translated right here. You see the SPLP, that sort of thing. Those are amino acids. So that tells you what gets translated from this little section up here. And I think, and I can't remember, and I'm tired. But anyway, you get it. You get it? Have I explained exons? Or have I failed?
Oh, yes, you've done a great job.
All I hear is crickets.
I really like this system you're using. What is this?
NCBI.
Pretty cool.
That is, well, in fact, I can, if you put this into your browser, I'm putting it in public chat, you should get exactly the same gene I'm looking at. Or you may not get exactly the same resolution. There was one.
Is this just a human? Is this just human?
This is human, yeah. Yeah. I think that was the choice I made.
Yeah, I see Homo right there.
Yeah, it's called GRCH38. I believe that is the most current human genome that they have. I think 37 was the last one or something like that. But there's a whole bunch of information you can get here. You can get, well, Demeter and I, Demeter did a really good job of explaining this in a previous video. If you go back about a month, and he explained how to set up different tracks and look at all the different features of the genome. And there are a lot of them. So
yeah, it's pretty cool.
Yep.
You get zoomed in like that, and you can see the sequence and everything. That's
right. But the other thing you'll find out is that creationists will then say, "Oh, in these introns are important to God, because sometimes there will be a little RNA, a little tiny bit of RNA that is transcribed in from the intron going backwards. So, like you're reading, like this reading sequence is actually this way, but you might find it reading the other way, and it creates a little bit of RNA, and that RNA becomes important in the cell to regulate things, right? So, I mean, there are sometimes there are parts of introns that are really important, and like this little part right here might create an RNA that turns out it's needed to regulate some other process, that sort of thing. So be careful for that. You're going to hear them talk about genomes being read forward and backwards and, this way and that way. And it is such a complicated code that only God could have wrote it.
So introns are important to God?
Oh, yeah. Oh, fuck yeah.
Wow.
God is.
That's why my favorite Bible verse is 2 Introns 4:3.
Yeah, it's 2 Introns 4:3.
Go look it up. It's in the Trump Bible.
Okay. That upside down one?
Yes, you have to be able to read upside down.
Yeah. 2 Introns 4:3, you said it was? I don't know how to write biblical verses like that, 4.3.
Yeah, 2 Introns 4:3.
Okay. I'll have to read that one later. I never think to look to the Bible to shore up my knowledge of genetics. So anyway, should do that more.
That's the problem with our country today, Speed.
I know. We don't teach that stuff in school, and we really need to.
I know. We really ought to.
Because we're heading for the fall, and it's getting worse and worse. Society is getting worse and worse and worse.
The downfall will be not getting our genetics from the Bible.
I mean, I wanted to get the mail the other day, and I had to step over dead body after dead body. I mean, it's falling apart out there. Society is falling apart. Immorality everywhere.
My genes
None
are
of-
breaking down
None
right
of them
now.
ever committed second introns to memory.
Yeah. What'd you say, Brainbuck?
So my genes are breaking down right now. I can feel them.
Oh, I can feel them too. Yeah, I can feel them breaking down to the point where I'm gonna have to take a nap right after this.
You know, when I was a boy, Mama told me not to sit too close to the TV. And now
Yeah.
we have microwaves and computers. So, yeah, our genes are probably getting all that radiation and breaking down.
Yep. Well, that's why I became an alcoholic, sitting too close to the TV. It isn't such a problem with the newer TVs, but the old TVs, man, they just went right into your brain with those X-rays. And
I thought you were going to say you became an alcoholic to try to preserve your genes and alcohol.
that too.
Yeah.
Well, that's one of the reasons why a lot of my friends are dead and I'm not, because I outdrank them. I have so many friends that are dead now that predicted I was going to be dead by 24. Fooled them. Ha ha.
You act like you got one foot in
You
the
go to their
grave.
funerals?
One at a time, gentlemen. Say that again.
Go ahead, Brainbug.
Oh, I just said you always act like you got one foot in the grave anyway, Speed.
Yeah, I know. Yeah, it's a little bit of a joke. It's like I was at a New Year's party. Me and my two sons spent New Year's Eve together, and I was over at his house. The other son, the one that moved out, or the one that I don't live with, at his house, and I was hauling a Cub Food box full of dip and plates and stuff we had brought over down the steps at 2 a.m. to go home, and I fell down backwards, and my son Dustin said, "Life insurance." That was all he had to say about me losing my balance and falling down, is life insurance. So I thought I had cancer for the last 2 weeks. So I went to the doctor a couple days ago, and I found out what I actually have is rocks in my head. Apparently, I've got vertigo because the little crystals in my inner ear have become displaced. But I'm a lot healthier than I imply. So
Have you tried using some healing crystals to balance that back out?
yeah, healing crystals, right? I had this doctor who never drank and smoked, and he exercised and he dieted all his life. And he was my doctor for 40 years. And he predicted that because I smoked 6 packs a day, I'd be dead by 55. And then he got kind of pissed off and irritated when I didn't die. And then he and I did a calcium score at 64. That tells you how much blockage you have in your arteries. And I totally kicked his ass. His was like 110 and mine was 45. And he was so pissed off.
Is he still alive?
He's still alive. Yeah, he's the same age as me. And
Speed, I don't know that I would want to go to a doctor that is disappointed that I'm still alive.
oh, I loved him. He actually did a whole lot of work to make sure to give me diagnostics. So he was really, he also loved to test drugs on me. Because I would tell him subjectively how the drug felt, and none of his other patients could do that. So it was pretty cool. Because I was in antidepressants and lithium and a bunch of shit when I was in my 40s. So he'd be like, "Okay, what does it feel like?" And I'd tell him like in detail, what it is subjectively to be on this drug. But I'd meet him in the hallway for our appointment, and he'd look at me and he'd say, "You're still alive."
You have to wait till you're in the 40s before in your 40s before your doctor will start testing drugs on you. Because my doctor never wants to test anything on me.
Oh my! No, he tested drugs on me when I was in my 30s too. He just loved me. He was that kind of a doctor. He kind of had a reputation for doing a little over prescribing. But he'd read about some new drugs and he was like, "Oh, God, Mike's coming in. I can test it on him. Yeah, something like that. Anyway, he retired. What a loss. I'm trying to find a doctor that will work with me like he worked with me. Because if you don't want to die of cancer, what you do is you get a doctor that will give you diagnostics. So anyway, so back to Andrew. What else, Andrew?
Can you guys keep talking for a few minutes? I'm clarifying something with Dr. Dan.
Oh, you're talking. Okay.
Sorry.
Well, maybe you could.
I mean, I can bring up a topic, and then we can you guys can talk about it, I guess.
Yeah.
I don't know. The discussion last night was kind of a similar thing, or a similar kind of topic, I guess. Like it like I said, kind of revolved around the nested hierarchies and stuff. So that was kind of pretty much it. Hold on, give me 1 second. Let's see.
So, Brainbug, what are some topics that you're getting ready to put on your channel?
Well, I just did the stream about because I just found the lightning bug, I guess you'd say, a firefly that pushed back the age of bioluminescence in beetles back into the mid Cretaceous now. So
I just did a stream or something.
Yep, in the Myanmar amber.
Interesting. So, where are the biggest deposits of amber at?
The Burmese amber is probably the biggest deposit. It's really problematic because they're finding all these breakthrough life forms that are showing, the diversification and specialization that that we're just now beginning to reach the surface on in this amber. But they're sending basically what is essentially slave children down 700-foot holes to dig for these.
What?
So it's there's some ethics to it too that
they're doing this now.
Yeah.
Oh my God.
Wait, what are they doing?
Sending slave children down 700-foot holes to.
The death was hyperbolic, just to be clear.
Yeah. Okay. Yeah.
Well, you may not be that far from the truth because I mean they get paid so little.
Yeah. The kids are slaves. I mean, they're paid pocket change, but I don't know exactly how deep they dig down.
Well, you remember that little girl that fell in a well? I think Jessica in Texas, and it took like 3 days to get her out.
Uh-huh.
They could have said, "Hey, kid, while you're down there, look around,
see if there's some amber."
Yeah, get back to us. Yeah, or oil. If you see any oil down there, let us know.
Anything of value, pirate treasure.
Yeah, if the kid comes up and it's greasy with oil, then you got a place to drill. Yeah, you just push the little suckers in the well.
Yeah. But it seems like not a month goes by I don't hear about something new that they've discovered in that amber. And I look at the articles and I read the papers and I feel guilty about it because I know that the only reason that we know about this animal is because some kid crawled down the hole for pocket change.
So you mean evolutionists were wrong about how long ago that evolved?
What's that?
So evolutionists were wrong about how long ago bioluminescence evolved in insects?
Yeah. It completely destroys the evolution model.
It did, yeah.
Oh, boy.
Yeah. Anytime anything changes, it destroys it.
I know I saw on PBS Eons last night that they discovered something called a hellfire ant.
Oh, yeah. Their jaws open up and down. They're not around anymore. They were Cretaceous ants that their jaws open vertically. So kind of like a closest thing you could think of. I could think of is like a rhinoceros beetle. How their horn is actually part of their jaws that they used to pinch. The ants had jaws like that. Really wicked looking suckers. I watched that Eons too. They were talking about why, and it goes into the kind of the evolution of eusocialism among arthropods, because they speculate that the way that they're finding them and the minimal amount they have, the diversity. I've read papers on them too, but they have the diversity between the males and females is more similar to what we see in like a velvet ant than what we see in like the true ants, Formica.
So this is a new species of ant they found.
Well, no, it's not a species. It's a whole clade of ants. Yeah, there's a few different genera in the clade. So
Why they talk about how there were signs of you called it eusociality, earlier than we had evidence of before.
Right.
Is that accurate, Brainbug?
Yeah. In the sexual dimorphism between the male and the female was less, but they were showing a trend toward it's not hellfire ants, it's just hell ants.
Hell ants,
okay. That's why I'm ending up with the ballad of the hellfire ant. I don't know what the hell that one is, but
that's Brainbug's favorite song.
Yeah.
Especially when he goes out honky tonking. He always has to have that on the jukebox.
Gotta get my lion dancing in for the year.
Exactly.
Yeah, look at that.
Oh wow, that is interesting.
I start to itch and have little burny feelings on my legs and my feet just by looking at pictures of ants.
Yeah.
So you look at their jaws and you see kind of a analogy with jawed tetrapods. In that you'll see the correlation there. It's a virgin evolution feature compared to most insects have horizontal closing jaws. So yeah.
Well, that is evolution.
Some different species have some pretty wicked horns and stuff on these hell ants.
Wow.
Man, I love looking at these in amber, and then the drawing that these people do of the interaction. Look at that.
Oh, cool.
Have you ever looked into Chimera arachne, the spider with the telson like a scorpion?
Is that they don't spin silk, do they? What is that one that it's an arachnid, and if any normal person would have looked at it and think that it was some kind of spider, but it's not.
Amblypygids. Hold on,
okay. Don't put any pictures up because I don't like looking at them. No, I'm gone.
Oh, okay. I got a taxidermied one behind me. I was gonna pop it up, but if you,
I prefer not to see it.
I don't want to trigger you. Yeah.
Yeah, I have I could probably stomach it, but it's weird. It's just this weird like thing that my body does when it senses that stuff.
Yeah, there we go. Now this is a fossil species of I believe it's Cretaceous too. Cretaceous, 100 million years ago, and it was like a sort of so the first arachnids come onto land were scorpions, and essentially spiders are descendant from scorpions. Amblypygids are, mites, everything. So this gives us an idea of what spiders looked like as they were in essentially a transitional state where they still had the remnants of the telson. The telson is the tail with the stinger on it. Now this animal here did not have, I guess he doesn't get pulled up anymore. It did not have venom that they can, from what they can tell, it didn't have venom in its telson. So it's just kind of a vestige for the species.
It's pretty cool.
Is there a difference between vestigial and vestige?
No, it's just tense, I guess.
Yeah. YouTube is cool because I can see myself in the past if I have a camera on.
Oh, and By the way, hi Danny, hi Demeter. Hello.
Hi, Demeter.
Hello.
I got everybody talking about bugs, so sorry.
I like bugs. I just can't. I used to have such an interest in them, and then I did a project on spider silk in high school, and something happened to where like I couldn't be around them anymore, or it was hard to be around them.
So is it real spiders, like true spiders, or like with tarantulas and stuff bother you too, or is it
all of it? Anything that has, I think it's something to do with the legs, but I try to like expose myself to a lot to kind of overcome it. It's gotten a little bit better. It's like, I guess it's like a legitimate kind of phobia.
Yeah.
And it's so inconvenient for movies. Just like because I'm actually fascinated by them. I actually I don't find them.
Well, that evolutionary response is, I mean, it's beneficial to you as a survive your species, to survive in your species, because if you have that aversion to leggy things, then you're less likely to get bit by a venomous.
There was a point where I blacked out. I was doing something with a fireplace and a big wolf spider, and I just blacked out right there. So I don't know. If we're thinking of it evolutionarily, I don't think that's beneficial.
No,
right?
Yeah, not if you pass out from it.
Yeah. And so I realized this is very inconvenient. So I tried to I started with pictures, and they'd be the size of a little thumbnail, right? And I tried to get as close to it as I could. And then doing that a lot really helped.
What about jumping spiders? They're adorable.
They're much better. Those are not near as bad. I think what I've realized is that it has something to do with the legs moving. So pictures are a little bit better, but oh my gosh, I think I would black out if I found something like a wolf spider on myself. I just don't. I can't even.
Do you live in the South?
Yeah.
Yeah.
Louisiana.
Okay. Yeah. So
but I don't really think there's going to be a good explanation, because I used to be fascinated by the things. I wanted to have pet spiders. I caught them, and I put them in jars, and I would try to feed them and stuff like that. I would stare at the windowsill when it was getting its prey, or when an insect flew into the web, and stuff like that. I loved it. And then I don't know what happened around age 15. It just took a spin. So my interest in them and the sort of alluring aspect is still there. I just can't. If I sense them in a way, I just can't handle it. My body can't handle it.
Was that about the time you watched Lord of the Rings? With Frodo and that up in the mountain fighting that big giant spider?
Because I've seen that movie so many times. So in the movies, I think I actually saw it. I had no problem. So this was, let's see, when did Return of the King was way before I was 15. I think 2004 was that Return of the King? Or 2003. So no, yeah, I was 15 in high school. That's for 2008 or maybe something like that.
Okay, so that make you a couple of years younger than me then.
Let's see.
Speed.
What?
The man is in the chat.
Oh no! Which man is that? Let me
The
look.
one standing there.
Oh my God, that guy! We had that guy in last night. I don't recommend it. I'm letting people in, Andrew. I'm such a pushover.
It's okay.
I mean, hey, I'm a nice guy, okay?
Yeah,
You're a nice guy, Midnight.
Hey, Midnight.
Hey.
yeah. I'm not going to tolerate any over talk. I'm going to become less tolerant on this channel. You guys will get removed to the back, to the back studio. If you get into any pedantic over shouting each other, I can't take it anymore.
Yeah, well, I'll be good. Just Danny isn't.
May I point out that there's a couple of questions in the chat for the meter.
Okay.
One about genetic entropy, and then there's another one a little bit later.
Who asked? Let's see. Who is asking? Standing for you converted in no time after. He thinks you'll be converted. Okay. Where am I at in terms of finding this? Okay. A question for Demeter from Immutable. Demeter?
Yep.
Are you with it?
So, are inorganic components something which is necessary for Luca to utilize in order to be self-sustained? Well, Luca is very advanced form by itself. It's already, if we are talking for Luca, according to everything we know about Luca, you're supposed already to have almost entire translational system in all of the basic metabolism undergoing. So Luca is already finished unicellular or contacting with many other unicellular organisms. So in that case, if any inorganic actually stuff around, probably will be important as it is important for any regular microorganism to exist. No much difference in that case. So Luca is very advanced, but if you're talking for the entire transition from Terra Darwinia to Luca, this is a huge, enormous evolution process. So just a simple complex, which is stabilized complex and capable of evolution, assuming start incorporate different metabolic pathways, and of course it depends on the environment. Everything will be more and more complicated. However, so the entire process from Terra Darwinia to Luca is a process of independence from the environment in one way or another. That's why getting more complicated. And every time when you have additional pathway, metabolic pathway, you have a step of independence. So I don't know how clear I was, but that's pretty much. Yeah, I'll be back after 2 minutes.
Okay. I think inorganic. Well, are you talking about magnesium, iron, sulfur, all of that? I think that was important very early on. I think that was an important part of it.
But don't they find a lot of that in meteorites?
What's that?
Don't they find a lot of the requisite elements or whatever in meteorites?
Yeah, they found a lot of amino acids, nucleic acids, a bunch of other things. But I think what Immutable is talking about, and I'm not entirely sure what he's talking, is basically magnesium, the minerals and how they interact with proteins, and they create these catalytic domains that end up doing some sort of chemical magic. They become an enzyme, or they make a reaction happen faster. And those, I think, were involved in very, very early prebiotic compounds, how life came up, basically. And that would be the next thing. Demeter's looking at nucleotides and amino acids, and basically that level. But there's also inorganic. There's a bit of geology involved in abiogenesis that we have. That it's going to take us forever to unlock because there's so many possibilities. But we all, all animals use minerals, magnesium and iron, and they're very important in catalytic functions in enzymes. So I'm babbled.
So Speed, since Demeter's here, you mind if I ask him about his thoughts on last night's discussion and what he thinks about a question I have for him?
Okay, no, go ahead when I'm not sure if he's back with us or not yet. But yeah, I got nothing today. I'm tired.
Sorry.
I copied Jerry Penna's question for Demeter into the private chat because it took me forever to find it. So I thought I'd save you the trouble.
Okay. Here's what Jerry asked, and there's no way to show private chat, so I'm going to put it in public chat again and display it.
Of course, I don't know. Somebody may have answered his question in chat, but
Quantum tunneling.
Oh, bacteria. Yeah, that would be natural. Are you with us, Demeter?
he said a couple minutes. So that was probably in quote.
Maybe give him a minute.
Yeah, yeah. Let's see. Jerry Penner stars create organic elements. True story. I'm not sure what you're talking about. SFT. I need to crash. It showed 2 or 3 hours. That hurts the way. Oh, Dessel Drace. I was calling him Dressel. Dessel Drace hasn't slept since last night. Just horrible. Julio. We have a Julio Grim Reaper. I don't know. And we have Stephanie. Interesting, Stephanie. Oh, here we go. Carbides, carbonates, some oxides of carbon. Cyanides are also inorganic. Yep. What's happening? Demeter doesn't know the concept of genetic entropy. Yeah, because
Because of the creationist thing.
Because it doesn't exist. That's
And
why.
the nearest thing there really is to it, Dr. Dan pretty much worked on in grad school or while getting his PhD or something like that. And he tried to create it, create a situation like it, and he just couldn't.
Hmm.
I don't know if he's still listening, but maybe he could clarify that. Or we could just go ahead and watch his several videos dealing with genetic entropy.
Who's
Okay,
that?
I'm
Dr.
back.
Dan?
Genetic
What are people
entropy.
saying about Waco?
Okay,
We should've
so
died already.
where was that question? God damn. Okay, so we had a question, Demeter. So Standing for Truth has a long time ago came up with this deal that bacteria mutate 3 orders of magnitude slower than most mammals, eukaryotes. And they're claiming that, one of the arguments against genetic entropy is that bacteria would have gone extinct 100 times over, to use Kondratyev's famous quote, except he used it on humans. And Standing's reaction is, oh no, they mutate real slowly. There's only one mutation, like roughly one mutation per copy, that sort of thing. And I told them that the reason for that was that bacterial genomes they don't have introns, they're far more conserved, as far as I know, and they're smaller usually. So do you have any comments on that or an answer to?
Well, you have to, and everybody should understand the evolutionary way of thinking. So I don't know, is the bacteria mutated faster than eukaryotic? I don't know that. I didn't measure.
They have much
So,
smaller generation times, but then they mutate a lot slower.
However, you have to pay a simple, everything it's very clear from evolutionary point of view. For example, in bacteria, if something is damaged and this is vital, the bacteria simply won't grow. That means exactly that you will left only with those mutations that will be tolerated and nothing else. The same is true in any organism. It doesn't matter is it bacterial or not. That's why in eukaryotic, for example, if you compare the conservative regions of eukaryotic genomes, for example, you will see very clearly that the most conservative regions are the most important actually for survival. And it doesn't matter how fast, it is matter, of course, but if you have some sort of regular mutations, which is absolutely true, you have such a regular mutations. We actually measure that very frequently in our laboratory, was measuring how it's mutated, how much mutation is accumulated. It's accelerating dramatically if some of the repair proteins is screwed up, for example, or something like that. You will see very clear picture that everything with the mutations will survive if the mutation it's not essential. Could be neutral mutation, could be just something very weak. I mean, not dramatic phenotype or so. So I don't know this the entire concept that you will mutate, and everything will go away after a while. It's just a linear fashion of not understanding of the evolutionary point of view. I mean, imagine if you have just one bacteria left in particular conditions, because all the rest of the bacteria is just died from, let me say, mutation rate, high mutation rate. This bacteria will overcome everything just in a few hours. So all longer, but it is just survive whatever it's not hit. I mean, the mutation will not hit the important functions, and that's why it is as it is. So examples are a lot. There are lots of examples in the animal kingdom or anywhere you want to touch. So I mean, from my perspective and from perspective of any evolutionary biologist, this is not even a problem, not even a question.
Yeah. Yeah, well, I think Standing last night said that Sanford made no prediction, or he did not predict that bacteria would go extinct. And I don't think he said they wouldn't either. But anyway, so some people have used bacteria as an argument against them because they have much smaller, shorter generation times. So obviously they are way ahead of us in the terms of the number of generations. And the idea is if genetic entropy is true, then they all should have died out by now. They should have accumulated mutations. But Standing for Truth has a bunch of rescue devices for that. So,
but in fact, you have to know that this is not linear phenomenon. You may have very small mutation in the organism to die. Very little, just even point mutation might kill the organism sometime. Or alternatively, you may have a huge chunk of DNA that is mutated and nothing specifically to happen. So it's nothing like linear fashion about that.
Well, they just want to cherry pick a bunch of little one-off things to try to make their point, and their point just doesn't stand overall because for the most part, they don't understand purifying selection, how it might work at all, or they don't want to talk about it.
Yeah.
They insist that the neutral theory, they insist, doesn't work for purifying selection. And it doesn't make any sense.
So immutable density, do peptides have the chemical activity necessary to fixate biologically relevant metals to their proper oxidation stage for reactions and after catalyzing some reaction? So some reactions are independent from peptides, and that's for sure. But peptide, I mean, definitely you have to, in order to understand the entire process, and I explained many times this particular way. When you have short peptide, the peptide can do whatever the amino acids are inside the peptide. So let me say, if you have two aspartic acids in the short peptide, like for example, 10 amino acids peptide, and you have two aspartic acids, which is a negatively charged amino acids. Those two aspartic acids will attract magnesium ions or any positive ions and will hold it. That might be important for some reactions. So it's not like you have peptide and nothing can happen. The peptide can be involved in some simple reactions with the metal catalyzed reactions. But having a little bit of effect, could be very advantageous in the beginning of life. Just a little bit over assuming some metabolic reactions that is coming more frequently into the system because of the short peptide, that could advance the system a lot. And of course, once you have longer peptide, now you have a folding process. The folding process is something which is actually drives dramatically the evolution, because longer peptide and folding creates a new functions, can create a new catalytic centers. Once you have the catalytic centers, your functions became unlimited. So you can do whatever you want in different mutations, different combinations. And also, it is assumed that the proteins that use catalytic center with some sort of catalytic substrates like metal ions or NAD or fat or different kinds of those kinds of cofactors, those actually it shows and demonstrates the evolution very clearly, because it's not perfect. So you don't have perfect catalytic center created only by peptides, only by proteins. So you need the help from some sort of adapter to do the job. And that's very important. Actually, you can see that evolutionary how it goes. As an example, I can say, if you the RNase is an enzyme that digests the RNA. It's a very simple enzyme and digests RNA without help of any cofactors. That's interesting because DNases that digest DNA actually require magnesium as a factor. So here is the point. Obviously, RNase, it's just a little bit more ancient protein RNase than the DNases. It's a newer proteins, evolutionary speaking. Therefore, it's a very interesting phenomenon how the cofactors comes and helps for different reactions. Of course, this is enormous, a huge kind of field of information and study. You can bury yourself in those kinds of study with so many reactions and information. You can, it's enormous area of expertise. I don't know. Immutable, are you happy with this answer or not? So, any other questions?
I was kind of getting into thinking about how big peptides have to be before they start creating these active domains, these folds that will wrap up an ion and do something.
So I can tell you there is no specific boundary, because even the very short peptide is kind of curvy, could be slightly curvy. But in order to bring more variety, you are seeking for something that it's beyond 20 nucleotides. 20 amino acids. So
yeah, that's
something like that. And as go further and further, became longer and longer, the situation, of course, became more complicated, and catalytic center could be more with high variety. Not to mention that you have also sometimes functions with making complexes between the two different proteins. So the actual catalytic center is formed by combination of several different proteins, and that's also it's appeared later on. For example, if you take MCM complex, today modern MCM complex of eukaryotic system, which I was studied very long time. It's an absolutely important helicase for the helicase activity of the DNA replication. But the MCM complex is contains 6 different MCMs, different MCMs proteins, which actually function as a helicase only when they are together, all of it. So, and there's hundreds of examples. This is just what came in my mind, but there's much more examples. So immutable sounds, he's happy with the answer. Okay.
Good. Yeah, and back to bacteria and the genetic entropy thing. It's the silly idea that I mean, we could look at insects too instead of looking at bacteria and ask why are they not extinct? Because Drosophila, I think they have like 20 generations a year or something like that, or 24 in the wild. In the lab, they have far more than that, I think, or something. But I mean, why are they not extinct? You know, why hasn't it? And I think the Drosophila mutation rate is pretty similar to mammals. So we could look at that one, you know, and talk about that. Standing has a standard answer for bacteria about how they don't mutate as fast and all this bullshit. But let's look at insects. They always want to talk about human beings.
It doesn't make sense bacteria to mutate less or further or more or less. It very frequently the bacteria is exposed in the very harsh environment. However, they do have a very good repair systems as well. So not to forget, you should not forget that the mutation, it is not recognizable anymore from the repair system, because mutation is already part of the genome. So it's already happened. But before to happen, you have a process of repair. So you have a long process of repair. It doesn't matter, is it bacteria or is it in eukaryotic cells. The result of not very good repair could be actually mutation. But you should not forget that the repair mechanisms are very good of doing that.
I put most of the things that we've talked about today in the description, also links to all that stuff like the hell ant and all the other things. So if anybody needs to find those things again, and this is just a little thing I brought up on the MCM complex.
MCM complex, oh my goodness.
So are you familiar with Ming Li? Or
What Ming Li?
the one who wrote the paper?
No, there's lots of papers on MCM. This is it's actually a very old one.
Yeah, this one seems like a more generic explaining it or something. I'm not sure. Does it have a... Anyway.
I mean, the MCM, I gave MCM just as an example of cooperation between the different enzymes, different proteins. But that's just one of hundreds of examples. So it's nothing specific for showing here.
Yeah, I just like to throw links up.
Oh, yeah, I'm not lying. For sure. Guarantee you.
Somebody said they're lying or what?
So, Demeter, if I may I ask you a question about molecular phylogenetics related to my discussion with SFT last night?
I don't know. I haven't heard about it. So what exactly you refer?
Oh,
but you can explain, yeah.
Well, like my question would be, so last night SFT claimed that evolutionary phylogenetics assumes genealogical relatedness in the past a priori before it builds phylogenetic trees. And Dr. Dan Creation Myths in the chat, for instance, said that this is true, but that it but that based on that, if we assume that is true, we can make models and test them and make predictions that work. If we assume shared history, basically. So I want to ask you what your thoughts are on this in terms of molecular phylogenetics, and if it's true that in phylogenetics we assume a shared history. I guess I'll start off with that first.
I mean, in my mind, there is no doubt that phylogenetics is showing shared history, and there is no. I mean, I don't see what the difference is. Of course, sometimes the phylogenetics gets complicated. Sometimes you have also a horizontal transfer of genes. And that can complicate the just building up the proper tree of phylogenetics. Sometimes you have a kind of synonymous structures, like you have the same function, the same structure, which actually is built up from different sequences of amino acids, and that's very interesting. It's like sometimes you have the same function appeared several times in the evolution. Sometimes, for example, the helicase activity that I mentioned. Sometimes you can have helicase activity from non-related proteins. So, although it's absolutely true, everybody should pay attention. There is some details that can complicate the situation. For example, sometimes proteins like aminoacyl synthetases, which are related for sure, the phylogenetics is very difficult just based on a sequence, because in order to recognize 20 amino acids and all of this complicated genetic code to be recognized well, the ancestors of aminoacyl synthetases needed to diverse a lot. To some extent, the diversion of the sequence became so extensive that you have difficulty to recognize the common ancestor. This is the reason why we know about the LUCA, and actually we know that the LUCA is approximately about between 300 plus minus number of genes, because if you try to make a phylogenetics of all of the genes from all the kingdoms, from all of the biological world, and you come to the common genes. That's why we know that LUCA did exist in the past, because I mean, the genetic code is one thing, a very similar, almost identical translational system, and all of that. So whatever Darwin was thinking about, we actually prove it now just by phylogenetics. I mean, that's my thinking. So just be careful of the kind of the differences in the nature. Sometimes nature is laughing at us because the processes are very different. As I said, you may have a gene from mosquito, although you may not have at all any ancestors with mosquito. Let me put this way. But how that might happen? It happened by horizontal transfer of genes, simply by virus transmissions could happen. So it's more complicated, just linear fashioned way.
So Standing for Truth in the chat is basically claiming that Dr. Dan Creation Myths doesn't know anything about genetics, and he showed his field of expertise, which is an auto mechanic apparently, or maybe a consumer or a suburban consumer who uses a car, and he's always using car analogies. So he's trying to have a genetics discussion with Dr. Dan, claiming Dr. Dan doesn't know anything, and I'm sure he'd say the same thing about the rest of us. But his example is taking the emptying the stuff out of his trunk to get better gas mileage. So I mean, he's got a hell of an argument there. You got to admit.
Even the cars arguments are very easy to work with, and I'm not afraid of analogies.
Yeah, well, I get so tired of car analogies. We were talking earlier about doing a phylogeny on cars. You know, get all the current models of cars and trying to run them through some algorithm to create phylogenetic trees for cars and see what comes up.
Yeah, because you have selection, right?
Yeah,
there are lots of cars that was created from creator human being, like exotic car with some kind of. I remember there was once a car created with some sort of steam engine, or there was a cars with even rocket engines. There was a cars with all kinds of turbine engines, all kinds of those. And none of those cars actually are on the road today.
Never mind.
Is that what you learned, Scott?
Yeah, I was just gonna let you know Dan wanted to come in.
Yeah, I just spotted that. So Andrew's got it too. But
yeah, I sent him a link.
Not emptying the trunk, removing the doors, removing the seat. Yeah, okay.
Actually, that's happened with the parasites.
Dan, how's it
Hi,
going?
Dr. Dan. Hi, Dr. Dan.
Hi, Dr. Dan.
So Dr. Dan, I apologize if I misunderstood what you were saying earlier.
Oh, no problem. It's just that there's so on that issue, there's just the colloquial definition of assume versus we're testing a hypothesis definition. And I want to make sure we're all clear on that when you're testing a hypothesis like universal common ancestry, you know, in terms of your logical flow, you have everything shares a common ancestor as a premise. So that's an assumption in the kind of technical sense, but it's not like I'm just assuming it for no reason. Like it's the basis for your predictions. But the thing, if I can have a couple minutes, because we're talking about this bacteria thing with genetic entropy, and I can explain that real quick if everybody wouldn't mind, because it's a really simple reason why bacteria should be susceptible to genetic entropy if Sanford is correct. And Speed and everybody else, would that be okay? I can only stay for like maybe 10 minutes.
Yeah, go ahead.
Like,
Yeah, we're wrapping up in about 15 minutes anyway, so go ahead.
It's like this. So what Sanford claims, if you go back and read Sanford's book, Genetic Entropy and the Mystery of the Genome, the idea is that if you look at all the possible mutations that can affect a genome, the vast majority are harmful. And if you tally up all the effects, the net effect of all of those mutations is harmful. So it doesn't matter if bacteria have, like, if they're simpler or if they have a slower mutation rate or whatever. What matters is, are all those mutations actually happening out of that spectrum? And bacteria have really fast generation times and really big populations. Plus, compared to eukaryotes, much smaller genomes. So you put that stuff together, and within a bacterial population, you're sampling every potential mutation. There's going to be no members of that population that are without any mutations. Now, within a specific generation, you might get lucky because sometimes the mutation rates are as low as 1 mutation per 10 million bases, and some of the genomes are smaller than that. So you could get a single, binary fission event where there's no new mutations in there. But that's not going to happen thousands of generations in a row. Within every lineage within that population, you're going to have mutations occurring. And because the populations are so big, you're going to sample every potential mutation. Now, if Sanford is correct, that means that entire population is going to be getting worse and worse and worse over generations. And because the net effect of all those mutations is to hurt the ability of those bacteria to survive and reproduce, then there's no way you're ever going to find a genotype that fixes them, and eventually that population is going to have to go extinct. Now, that's just following the basic population genetics as Sanford lays them out. Now, I see standing in the chat is saying I'm wrong. His responses so far have been that bacteria do degenerate, which seems to be, I mean, they would if genetic entropy was true. They should be. That's what I'm saying. And two, that they're simpler, and also that they're unicellular. None of that stuff matters. It's a question of generation time, mutation rate, genome size, and population size. Those are the 4 factors. And as I just described it, the way those 4 factors work, you sample the entire mutation spectrum within bacterial populations. That means there's no way for natural selection to save that population if Sanford is correct. And the nice thing about bacteria is we can get population sizes that are that big to test this in the lab to see if they actually do go extinct from genetic entropy. And I'm sure it's not a surprise to anybody, but when we do that, do they actually go extinct? No. That means genetic entropy is false. Like, it's that simple.
So
It's
when
not more
we
complicated than that.
start talking about genetic entropy this or genetic entropy that, if I say that's not a thing, I'm scientifically accurate. Is that correct?
Genetic entropy is not a thing. It simply
Okay.
does not exist.
Not
Like
a thing.
100%,
Right.
it doesn't exist.
So, Standing for Truth is asking how many cells do bacteria have, and I suggest that they have millions, depending on how you look at what is the organism, like what is the.
I mean, it doesn't matter though. It doesn't matter. They could be unicellular, they could be multicellular. It doesn't matter, because what matters is within the population, all those individuals, is there anybody that has a genotype that is getting better? And according to Sanford, the answer should be no.
Sanford doesn't take you seriously.
Sanford doesn't even know my. Actually, he does know my name because he wrote that he co-authored that piece with Paul Price and Rob Carter. And no, he doesn't take me seriously, but that's fine.
So, Danny, Dr. Dan, as I see, you are lazy as me because you are not shaved as me. So,
This is not a pandemic beard. This has been for years and years. Everyone else is just catching up.
I'm just lazy sometimes to shave myself. That's why I didn't shave. Simply, what the heck.
Can I ask, because I see both Standing and Lena Powell in the chat saying Sanford explains this. So what does he explain about bacteria that makes them immune to it?
I've never seen him explain bacteria.
What's Sanford's argument about bacteria that makes them? Because Sanford very clearly says viruses are susceptible, right? And he says mammals are susceptible.
Yeah.
Right. He has the two extremes, but then in the middle you've got bacteria. It's like, well, why aren't they susceptible? And I've actually I've been asking Standing in the chat for like the last 20 minutes on and off, like what is the argument? And he's just giving me attitude instead of answering the question. But maybe Lena can answer the question, like, what is Sanford's argument for why bacteria can escape this mathematical inevitability when organisms that are smaller and simpler have smaller genomes, and organisms that are larger, multicellular, more complex, have larger genomes, are susceptible on either end of that? What's the argument there?
I have a question. Where can I find 10 or 12 papers from that Sanford has published in peer review on genetic entropy? How many papers are published on GE?
So Standing, what's the bacteria what's the argument for bacteria being immune to this? Like, what is the math that show, like, explain why what I just said is wrong, instead of saying, oh, Sanford says that's wrong. Sanford doesn't take you seriously. Like, if it's that easy, just give us the answer. Right? I think that would be useful information for everybody to have.
He has his little Gallup somewhere up in chat. I forget what it is. He had like 4 items.
No, he said it's because they're smaller and simpler and they're unicellular, and therefore selection, and they have a lower mutation rate, so selection works better in the large populations.
And there's lots of them.
As I just, like, the thing that I just described addresses that. Because it's about the total mutation saturation. It's not about any individual cell.
Yeah, correct me if I'm wrong. But the reason they have a low mutation rate is because of selection, and that basically the purifying selection
They have their per base per replication mutation rate is really low. They have very high fidelity DNA polymerases, and they have really efficient error correcting mechanisms. So they do a really good job at DNA replication.
Yeah, and
it's worth noting that that's actually a phenotype, that you do see situations where they can increase that when it's beneficial to sample more mutations. And the fact that this is called a mutator phenotype, it's where they actually basically turn off some of their DNA repair stuff, so they increase their mutation rate by a lot in some cases. If Sanford was right, you would never see mutator phenotypes win.
Right.
Because that would just cause them to go extinct faster. The fact that mutator phenotypes exist means Sanford is wrong. Like that's a direct
also
test.
I have to add two things more. First of all, experiments has been done millions of times when you increase the mutation rate dramatically in bacteria, for example. Yeah, and just of course they die. That's how we are trying.
You can kill them, sure.
To kill, and of course there is a limit, which is obviously they can tolerate, and this can be calculated pretty much for any organism. Where is the limit?
Yeah.
But as I mentioned, this is not linear fashion style. Sometimes the hits might be very small and very little. Sometimes the hits might be big, but not a little. And also, additional second thing I want to mention is the eukaryotic system. So don't forget, the eukaryotic genome has a double copies of everything. It's called homozygosity and heterozygosity. Why we have homozygosity and heterozygosity, and why we have sex?
Yeah.
See?
Yep.
There is some reason for this, and the reason is exactly the same thing: diversity. So
exactly, and if more diversity necessarily meant going extinct faster because all these new variants are going to sample are on net harmful, then there would be no benefit to mechanisms like that, because that would just accelerate the rate of extinction.
So
I'm still waiting for this counter argument here, but I don't know if standing left or something, but he got real quiet.
So Dr. Dan.
I'm sorry, go ahead.
Oh, no, go ahead, Scott.
Lena had said that she wanted to put together her argument carefully, so I think she's taking a little more time. At least that's the way I understood it from what she said in chat.
So I'm looking at Google Scholar. I'm trying to find Sanford's published papers.
So Dr. Dan,
Standing, don't give us a link to a video. I'm not watching a 3-hour stream with 12 minutes of content. If you have an answer, put your answer in the chat or come in the room and provide it. Like, don't play this, I'm going to link you a video. You can go find it yourself. Also, the video you linked was about mice, not bacteria. It was 10 minutes and it was about mice. Perhaps you linked the wrong video.
Yeah, the title says the existence of mice and bacteria refute genetic entropy. And it's Sal Cordoba, which I don't think he is an expert in that area.
I mean, I debated Sal on this, and he agreed with a lot of my critiques of genetic entropy, including the one I'm articulating right now.
Yeah, he actually admits that it might apply to humans because
look. It could be, that's fine. It's Rob Carter. Rob Carter's wrong about this too. Don't name drop. I don't care. Give me the arguments. Why is this such a hard question? If you have an argument that refutes what I'm saying, state the argument. Why is this hard?
When
was that the Carter video? Wait. Actually, I know what video that was. I've watched that video. Oh my God. That's the one where Carter says that there are E. coli right now that have no mutations because there are, in some cases, the mutation rate is 1 every 10 million, and the genome is like 4 million or whatever. It's that video, right, Standing? I bet it is. He says in this video that there are, is it the one where he's like in the woods or something? I'm just pulling it up just to confirm that it's the one that I've seen.
I've got it up on the screen, Dan.
Oh, okay. So in when Carter talks about this,
does the existence of mice and bacteria refute genetic entropy?
When what Carter says in this argument is there are bacteria right now that have the same genome as when they were created, because they can sometimes do a replication cycle with no mutations. But that is bonkers, because think about this. It's about a 1 in, I think it's about a 1 in 3 chance of that happens. Maybe it might be more. I forget the math, but let's say it's a 50-50 shot, which is more generous than it should be. But let's say it's 50-50, right? That a bacteria reproduces with 0 mutations, let's just say. Now, that leads to how many generations do you have from creation to now? 6,000 years if we're going with Yek, okay? And bacteria have really fast generation times, right? We could be really conservative and say a day. So what's 6,000 times 365? That number, whatever that is. So take 0.5, the probability of having no mutations, and raise that to the 6,000 times 365 power. The answer is 0. Carter is completely wrong on this. It's not only is he wrong, that math is completely bonkers. It's not even close. So yeah, the thing that he says of why bacteria don't apply, it's because they have such slow per generation per replication mutation rates. But then he does the math on it, and it's bonkers, because he claims that there are no bacteria. There could be bacteria right now that have the created genotype because they've gone through every replication since creation with no mutations.
So I have a question. Why would Standing for Truth come on here?
Oh, he came on last night. I'd have to kill him if he came back today.
Now Lena has a question for...
Okay, Lena's got an answer here. Okay, simpler genomes, high population size, and short generation, lower all... No, it's not. And Lena, as I said, the fact that they have such large populations and small genomes mean that they sample every mutation. Within a generation, you're going to have every possible mutation. Because you're talking the genome for like E. coli, I think it's like 4 million. Someone's going to correct me on that. I think it's like 4 million nucleotides. 4 million base pairs. Now, within a generation, if you have, say, 1 billion bacteria, which is a trivially small population of bacteria, you're going to have every possible point mutation within that one generation, right? So that means that everybody in that generation has gotten worse, according to Sanford, relative to the generation before it. Because again, on net, the effects of these mutations are harmful. So you do the next generation, they get worse. The next generation, they get worse. The next generation, they get worse. There's no way to fix it. Even with the slow mutation rates, the giant population size and the fast generation times are the death sentence for bacteria if Sanford is right. And that's just simple math. And nobody has shown me the math that shows that I'm wrong. And it's a mathematical argument. It's a population genetics argument. Just show the math. Any change to the simple genome will affect the reproduction. I mean, yeah, and so you're right. Any change should affect it because there's very little non-coding, very little junk DNA, yada, yada, yada. And according to Sanford, almost all of those changes will be harmful. Therefore, that population is inevitably getting worse generation after generation and should go extinct. This is using Sanford's own math.
Yeah, it should build up in the population.
Yep.
Like
This is, look, I'm just using Sanford's math here, like describing Standing, that's the point though. It doesn't, selection can't see it and remove it because everybody is getting bad. Everybody is getting worse over time. Again, if Sanford is correct, right? If you're saying that Sanford's distribution of fitness effects is wrong, you're right to say that because it is. And that's one of the problems with this argument. That's what must be the case in order for bacteria and anything else to be immune to genetic entropy. You're absolutely right. There is selection and it sees those changes and removes them. That means genetic entropy is false. Exactly.
By the way, hey guys. By the way,
I'm setting a timer for myself. I literally have 2 minutes.
Real quick, you guys. I just feel the need to point this out. The whole created heterozygosity model directly contradicts the genetic entropy model. Here is what
Yeah.
Dr. Jeanson says in his paper, and I'm going to quote directly, "To be sure, this is not a deistic hypothesis. Under the created heterozygosity natural processes model, God doesn't create and then abandon his creation. Rather, the model recognizes that God is actively involved in his creation, providentially upholding it to this day, and the model recognizes that God works via means, including via the environment and the natural processes that he supernaturally designs and upholds. So, if God is supernaturally upholding his creation, and if God is all-knowing and all-loving and all of that, why is he letting the genome deteriorate into
Right.
eventual extinction?" So,
And Lena, the ones that have accurate replication will survive. There's no way to get accurate replication over every generation, every one in a row. There's no way to do that. You can string one generation with accurate replication, maybe 2, maybe 3. You're not going to get 10. You're not going to get 100. You're not going to get 1,000. Eventually, they're going to go extinct again if Sanford is right. And David's point is spot on. It's directly contradictory with created heterozygosity. It's one or the other. You can't have both.
yeah, exactly. Jerry, someone should give David an honorary BS.
I agree.
I
A real one.
Yeah, I would rather get a real one. Well, I am going to get a real one in about 2 to 3 years.
I'll be out of here soon.
Yeah, real quick though. I just had a really good conversation with the Dapper Dino on my channel, so go check that out.
Oh.
So
Where? Do you have a link? Put a link up for us.
I can post a link. I'll put it in the private chat, and you can post it in the public. It was a really good conversation. Spoke about.
Do believe you have a wrench.
Yes. Hold on 1 second. Yeah. But anyway, also don't forget to check out.
That's me. I'm out of here, everybody.
All right. Bye, Dan.
Thanks for letting me hop on.
I'm about to get out of here too, so let's wrap up.
Now I'm going to over to you.
Was this everything you expected it to be, Andrew? Did I do it again?
I didn't get to ask Dr. Dan my question, unfortunately.
I thought you did. Oh no, you asked Demeter a question. I thought you got Dr. Dan. He was in a hurry.
A very simple thought. If a creationist are correct with the genetic entropy, so in that case, God created all the living creatures and set it up, all living creatures in a downfall, because eventually all single creature will diminish because of the genetic entropy, eventually down. So why God created all of the creatures just to stand for a few hundreds of thousands of years, and that's it?
Yeah, I think bacteria represent a highly sped up version of what's happening with mammals. You know, that we can see that it still works. Standing for Truth used to famously say, we have 100 mutations per generation. Therefore, every generation we're adding 700 billion mutations to the gene pool, which is just such a complete misunderstanding.
As I said, every single time I have to explain, you don't have linear effect. You don't have linear effect. That's first thing. And the second, even one survival will continue.
Right.
So it is completely nonsense.
Yeah, with with 6 billion nucleotides, if we were adding 700 billion to our generation every time, every generation, we'd probably have tentacles by now. I don't know.
I even don't want to calculate those rubbish things.
Yeah.
I mean, why should I even try to do that?
Yeah.
Because calculating is fun.
Yeah, exactly. Okay, guys, I also gotta go.
Yep, let's wrap it up.
Come on. By the way, Skeet, can you hang on after you end?
Yeah, and I'm gonna do my my dramatic outro. Goodbye.
Before leaving, I wanna set up for Danny. Danny, hello, Danny.
Hello, Danny.
Me? Yeah, what's up?
So I want you, from a philosophical perspective, to think about what is intelligent. from philosophical perspective. And is intelligent is always mean that there is a intelligent creature behind. So I don't want you to answer me, Rao. We don't want to go in discussions, but that's an interesting question.
Well, I need to clarify what you mean. When I say that like someone's smart or intelligent, that's one meaning of intelligence, and then there's intelligence in the intentional sense, right?
As
And
a process. I mean, I in my mind, my question is as a process.
a process. Okay, I see.
Yeah.
Yeah, go think
So-
about it, and we'll have a discussion another time when everybody has more time. So,
All right.
okay,
Bye, guys.
okay, goodbye.
Bye.
I'm ending the broadcast. Goodbye, cruel world. It is so cruel.
Speed,
the
will
one-
you stop talking like that? Holy shit, you're starting to worry me.
I'm being facetious, and now that we have a new president, I'm so excited every day to listen to press conferences. It's just, I am so excited about life.
Like they're letting adults back in the room again.
Yeah. Anyway, ending broadcast.